Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Quick note before we begin, the Finding Genius Foundation, as part of the Finding Genius Podcast, has recently completed a book about understanding viruses. So the creation of this book was to interview 100 virologists, ask them a lot of deep, difficult questions, take the most difficult questions, and then re-interview the top 25 or so and ask them the hardest questions I could think of. And we compiled that all into a book. So you'll see question and 4 or 5 experts' answers. Question, 4 or 5 experts' answers. There's about 30 questions in the book. I think it's a great read for the layperson and for the researcher. It talks about a lot of speculation in the world of viruses, such as are they alive or not, and why is it important? Why do viruses go latent or hidden or ineffective or sit in a person or an animal or another creature for weeks, months, years, and then suddenly become virulent and affect that person? So there's a lot of really provocative questions in the book. It's now on Amazon. So if you go to Amazon and type in Finding Genius, you'll see the book on viruses. It's also on Kindle. The Audible version is in production and should be ready in approximately a month. But if you want to go and order it now, you can do so again by going to Amazon or Kindle, or go to findinggeniusfoundation.org and go to publications. There's an opportunity as well to get the transcripts of all the interviews and to hear the original interviews themselves. If we put them all together, the book would be about 1,000 pages, but we condensed them down to make it juicy and concise and tight and very interesting. So I hope you'll check out the book. We're now working on one about cancer, but this is going to be our goal is 3 times a year to come out with these masterclass books that I think will inspire new scientific research, and I hope you'll check it out. Thank you.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. Today my guest is Michael R Strand. We're going to talk about his research. He deals with parasitic wasp physiology, immunity, and what's called the polydNA virus. He's at the University of Georgia, and we're going to talk about his research. So, Mike, thanks for coming.
Thank you for having me.
Yeah, what got you interested in wasps and parasitic wasps?
It goes back a really long time. When I was in graduate school, this kind of interaction was in the early stages of discovery, and it just happened to be something that was going on in the lab in which I was a very early graduate student. And so that was my first exposure. And I stayed away from it for several years, but because of that exposure, I came back to it. And after I had a job, faculty position, the first faculty position I had was at the University of Wisconsin-Madison. That's where I started working with them.
What happens with a parasitic wasp? What does it prey on? What does it do? What's the basic life cycle of it?
So the basic life cycle, maybe we should back up just a little bit. These insects are incredibly common, so nobody really knows exactly, but the estimates are that maybe 10% of all the animals on the planet are parasitic wasps. So there's upwards of 1 million species of these kinds of animals. Nobody really knows for sure. And their life cycle, all of them have kind of the same common denominator. And so that consists of, if you remember all the Alien movies, Sigourney Weaver kind of stuff.
Yeah, the alien bursting out of people's chests.
Exactly. Yeah. That's pretty much, so the idea for Alien really came from somebody who had looked up the biology of parasitic wasps. That's pretty much what they do. So these insects are called parasitoids properly because of the suffix is parasite-like. And the reason they're referred to as parasitoids or being parasite-like is because they're free-living insects as adults. And females lay their eggs in or on the bodies of other arthropods, mostly other insects, but some other insect-like invertebrates are hosts as well. Most species are really specific. That is, they parasitize a particular species of host or a very small number of hosts. And so they lay their eggs in the body of that host, and the progeny hatch from that egg, and they consume the host. And then they ultimately emerge from that host to become an adult, which repeats the cycle. Most species are dioecious, so in the sense that they're males and females, and they have to mate. In order they mate, but males don't parasitize things. Only females do, because they're the only ones who can lay eggs into the bodies of host insects.
Yeah, I thought there was just one kind of parasitic wasp, but it was unique. But you said there could be a million different species of them?
Yes, nobody really knows, but in terms of described species, it's a couple 100,000, but estimates are that it's substantially larger than that. Virtually every insect on the planet is parasitized by some parasitoid wasp. And because most of them are really specialized, it just means that there are complexes of these parasitoids. That's why they're so huge and why they're so ubiquitous on this planet. They're just so tiny. Most of these insects are very, very small, and so people just are unaware of them.
Wow. So again, the mechanism is the wasp will find its prey. It's very specialized. It'll sting it.
Yep.
And what that paralyzes the prey, it lays eggs in it, and then
Yep.
what the prey recovers and thinks it's okay, but yet the eggs are growing inside of it, or does it remain paralyzed until the eggs, hatch and eat the animal?
Yeah. So some parasitoids do paralyze their hosts, but the vast majority of them actually don't. And so you can imagine with the diversity of that I'm talking about, that variations on the theme of laying eggs in hosts and developing takes on all kinds of crazy extremes. So many of these species are not only are they really specific to a particular insect, they're very specific to a particular stage of that insect. So some parasitoids only parasitize the egg stage of a host. Others parasitize the juvenile stages. A lot of insects undergo what we call a true metamorphosis, and kind of like a butterfly becoming a chrysalis, becoming an adult butterfly. That chrysalis stage or pupal stage, many parasitoids just specifically attack that pupal stage or the adult stage. Some of them paralyze their hosts, others don't. Some just feed on blood. Some just completely eviscerate and eat everything inside of the host. Some develop in a very particular organ. Some end up consuming everything, like I said. Some produce a single parasitoid from a host. Some lay many eggs into a host, and so they produce many progeny. And you end up with a giant emergence of wasps from the same host. And then a few super crazy ones are polyembryonic, and so they will lay a single egg in a host, but the egg clones itself inside the body of the host and produces thousands of genetically identical wasps that consume that host and emerge. So, and then some of the parasitoids are parasitoids of parasitoids. So they lay their eggs inside of a parasitoid that's inside the body of the primary host. And there are secondary parasitoids. And so like Russian dolls. So you end up with such incredible diversity in the biology of these insects that it's really kind of hard to wrap your head around it.
Yeah, I'm interested in what you just said. So there's secondary ones. So how many, what's the most extreme case of parasites parasiting parasites?
Yeah.
You've observed? How many?
Yeah, in the literature, the most extreme cases that I know of are 4 to 5 trophic levels. So a parasitoid of a parasitoid of a parasitoid of a parasitoid of a parasitoid. And it's a little bit of a kind of law of energy conservation. It's not really the efficiency of acquisition of resources as you go from one trophic level to the next is not 100%. So things just get smaller and smaller and smaller. It's not sustainable at a certain stage. So that sort of 4th trophic level is the most extreme that is known in the literature that's well documented.
So what happens in a situation like that? What happens to the intermediate parasitoids? Is there a staged feeding where the first one hatches, it eats the original host. Then when that grows fat enough, it activates the next one and the next one and the next one, or do they all go at once? Some of them find nothing, or what happens?
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
Again, as you can imagine on these sorts of things that in some cases, it's pretty much everything all the way down the trophic chain ends up dying except for the last one.
Huh.
And in other ones, that's not the case. And so the host might survive, and only the parasite inside of the host ends up being killed. So, yeah, it goes in lots of different ways.
Huh.
And so, again, this is really quite common. Many parasitoids are attacked by these parasitoids of parasitoids, which properly in the literature are referred to as hyperparasitoids.
So when this situation happens, what's the order of the parasitization?
Yeah, usually the parasitoid of the primary host, the other type of insect, is there first. And then the hyperparasitoid is capable of recognizing hosts that contain the parasitoid that is the host of the hyperparasitoid.
Well, this could be very useful in stopping this. If you get a hyperparasitoid and you see how it works and what it preys on, I mean, I guess you could deliberately attract that so it could eat the parasitoid and maybe save the host.
If you don't die in the process, that's a real possibility. As you can imagine, one of the reasons that there's a pretty large and old literature about these insects, even if the public's not real aware of them, is that many of these parasitoids, not the hypers, but I'll come back to hypers in a second. Many of these parasitoids, their hosts are extremely important agriculture and forestry pests, and so for a very, very long time, certain species of parasitoids have been used as management tools for the control of pest insects, and this is referred to in the literature as biological control. And so you're using these insects, these parasitoids, as a tool in pest management in lieu of using an insecticide or using some other kind of control tactic. And so for a number of our major pest species, they're not pest species at all because of extremely successful biological control programs that have taken them off the table of having to be managed by use of insecticides or other tools. When biological control programs are highly successful, they don't require any additional input. The parasitoid was introduced to control the pest, and when completely successful, it's self-sustaining. So they drive the population of the pest insect down to very, very low levels where they're not causing significant economic damage anymore, but they persist through time. And so the parasitoid is just always there, and the host is always there, but at a much lower population level. And the grower or the manager of the product or the crop enjoys the benefits of this at an extremely low cost. It's kind of like immunization.
How many levels are necessary to protect one's crops, let's say? What if you just go one level with one hyper? Is that as effective as, 2 levels of hyper? Has anyone tried that?
Yes, is that all of our great biological control agents are what we call the primary parasitoids, the parasitoid that's attacking the pest. The reason I said I'll come back to hypers is that sometimes hypers can disrupt biological control. So if you ended up with a hyperparasitoid that ended up attacking the primary parasitoid that was the biological control agent, you can end up reducing its efficacy greatly or interfering with it being able to serve the purpose that was intended as a biological control agent. So in that sense, the hyper is a negative. Because
are hyper events rarer than primaries?
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Well, the hyper prevents the primary parasitoid from being able to reach population levels to be able to control the pest insect that's the target for control.
No, I know, but I'm saying like is it a rarer event? Is it common it sounds like it's very common for, a primary creature to be parasitized by a parasitoid, but
sometimes a lot of biological control programs, many of the pest species all over the world are introduced. So we have some pest species in agriculture and forestry that are native, that is, they were endemic to an area. But more often than not, most of our pest species are introduced from a different part of the world. And most biological control programs that are looking for a permanent establishment of the biological control agent, the traditional tactic is to go to the native range of the pest insect. So let's say, for example, the pest species has been introduced into the United States. People determine that let's say its country of origin was, let's say, somewhere in Asia. So then usually in its country of origin or region of origin, this isn't even a pest because parasitoids or other predators or parasites are keeping this thing at a very low density. But when it gets introduced into a novel region, like being introduced into the U.S., then those things that are maintaining its population at a very low level are no longer there. So people go to the native range seeking to identify what those things are. And so they frequently will introduce primary parasitoid or several primary parasitoids that are candidates for controlling this introduced pest. And in the process, they're screening quite carefully to not introduce any hyperparasitoid that's in that native range. So they're trying to prevent that from occurring. So because so many of our pest species are introduced, and because of tactics for trying to screen out or avoid introducing a hyperparasitoid, this reduces the risk of accidentally introducing a hyperparasitoid that could disrupt a biological control program.
So what's been observed from parasitoids and hypers? Like, what are some of the mechanisms they use to identify, this host parasitoid mixture and to infect it?
Well, what people traditionally do early on is they would have the primary host species, let's say the pest insect, and they literally go on expeditions to the native range, and they seek to collect this pest insect in its native range, and they literally rear them. And from rearing these insects, that is, bringing them into a laboratory, they parasitoids emerge from them. And then an exercise is put into place to identify what they are. Sometimes they're known, sometimes they're novel. They haven't been described by anybody before. And from this, people will then also try to learn a bit about the biology of, what stage of the host are they attacking, and how is it that they perhaps locate hosts, and where are they common and where are they rare, ultimately to try to make decisions about identifying candidates that show potential for potentially being able to be used as a biological control agent. And in the process of all that, sometimes hyperparasitoids are discovered as well, or hypers of hypers are occasionally discovered, and so on. So it's kind of building a story from a simple observation. And then there, like myself, there are people who over a long period of time have studied certain parasitoids and their hosts to understand, how is it that they kill the host, and how do they manipulate host biology, and how do they complete their life cycle? And so These parasitoids sometimes change the biology of their host greatly. Others are pretty crude about the way they kill hosts. They literally just eat them, and that's about the size of it. In this podcast, some of it was, part of the title was about Viruses. Viruses are just another kind of crazy variant on parasitoid biology, where one particular group of parasitoids evolved in association with a virus. And this association over evolutionary time has changed greatly. But in effect, the parasitoid is using an ancestral virus to be able to successfully parasitize the host that it develops in. So it's in a way sort of formed an association with an ancestral virus to be able to successfully develop in its hosts. So in a way,
what do you mean? How do you know there's an association between the parasitoid and a virus?
Well, the way it was discovered is that about in the late 1960s and early 1970s, people working with a particular group of parasitoids that are in a family, two different families of wasps that are called, the family is Ichneumonidae and Braconidae. These are just huge. There are many, many different families of parasitoid wasps, and those are two especially large ones. And so what was being done at the time was people were looking at these wasps, and they were doing some work by electron microscopy, looking at the reproductive systems of these wasps. And when they looked in the ovaries of these female wasps, inside of some of the cells of those ovaries contained what looked like virus particles. And when they looked in the reproductive tract of the wasp, they noticed these virus particles were literally accumulating in the lumen, that is in the reproductive tract where the eggs of the wasp were. And from this, they ultimately determined that these particles contain DNA. And so they not only looked like viruses, but they contained a nucleic acid that would be consistent with also being a virus. And then they finally discovered that when a wasp lays its egg into the body of its host, these are all primary parasitoids, they inject a quantity of these particles into the host as well. And these particles infect different cells of the host. And then from this, people learned, including myself, that the DNA in these particles, that there were many different genes that these particles were delivering to host cells. And these genes encoded for products that changed the biology of the host in ways that enabled the wasp progeny to successfully develop. And so the notion got established that these wasps required these virus particles to infect their hosts in order to be able to successfully parasitize them, in order for their progeny to survive. And then from this, a lot of details began to emerge about where did these things come from and what was their evolutionary history. And ultimately discovering that in a way, they're not exactly, maybe in a technical sense, viruses anymore. That they evolved from a virus, but they've actually are parts of viruses that now have become part of the genome of the wasp carrier, which I know sounds a little complicated, but that's actually kind of what happened, and I can explain it.
They become part of which genome, of the parasitoid or of the original host?
Of the parasitoid. So if you want, I can take a couple minutes to kind of explain what happened, or what appears to have happened.
Sure.
Different types of molecular biologists and systematists could take information from the DNA in those virus particles. And from this, you could use it to discern what they came from. And for the ones that are associated with braconid wasps, it turned out that evidence suggested that these particles are being produced. They're related to a family of viruses that are called Nudi Viruses. And Nudi Viruses are a group of viruses that infect many different types of insects. And so by taking some other pieces of information, it looked like that some kind of ancestral Nudi virus became associated with the common ancestor of braconid wasps about 100 million years ago. So very, very old. And then some really remarkable changes occurred. So for a regular virus, like a Nudi virus or pretty much all major viruses, the virus particle ends up packaging the genome of the virus. That is, all of the genetic components that are necessary for the virus to be able to replicate and propagate itself in its host. And all viruses need some kind of other host organism in order to be able to maintain itself. So the host could be a bacterium, the host could be an animal, the host could be a plant. We have plant viruses, animal viruses, prokaryotic viruses. There's zillions and endless numbers of different types of viruses. So viruses can't propagate themselves independently. They need a host cell to be able to do that. But the particle for virtually all viruses contains all of the genetic information to do that. When people started studying the virus particles that were in the wasps, one of the discoveries is that they contained DNA, but none of the genes that were in those particles matched known genes that have functions in particle replication, that is, in a virus being able to propagate itself. And then through studies of the genomes of the wasps, it was discovered that many viral genes were now integrated in the genome of the wasp, and they were all genes that were related to Nudi virus replication. So what ended up happening is the ancestral Nudi virus became integrated into the genome of the wasp, but only part of that genome of the Nudi virus ends up in the particles that replicate in the wasp reproductive tract. So the particles are no longer a functional virus exactly. What they become is kind of like a gene vector. So the particle is a means by which genes can be delivered to a host insect, but that particle itself can't propagate itself in the host. So instead, all the replication machinery is part of the wasp. And so the virus, in a way, has become this. What wasps have done is they've taken Nudi viruses, they've taken the ancestral Nudi virus, and they make Nudi virus-like particles, but they package very specialized genes that are important for changing the biology of the hosts that they attack. And all the replication machinery just stays inside of the genome of the wasp. And so the whole thing is just vertically transmitted. And the reason I said it's not exactly a virus anymore is because it produces replication defective particles that the wasp uses like a biological weapon to attack host insects.
I don't know if the virus is doing anything, but
the virus is
the host that the virus interacts with is able to strip off useful pieces of its RNA or DNA to use.
Yeah, so the wasp has all of the parts of the virus in its own genome. Everything is integrated into the genome of the wasp. So all the viral genes have essentially become wasp genes. Mm-hmm. And this machinery, though, inside of the wasp, in cells in the ovaries of female wasps, get turned on to make particles that can package genes that get put into those particles. And these particles the wasp can inject into a host insect, which is usually the caterpillar of a moth. And these particles can then deliver those genes into the cells of that host insect and express all kinds of things that this wasp egg that also got put into the caterpillar need in order to be able to successfully develop. But every single gene is still inside the genome of the wasp. It's inside the mother who's making these particles, and it's inside the wasp egg that got injected into the caterpillar. So all of these genes are essentially just part of the wasp now, and they just get transmitted vertically one generation to another. And this is how this thing has been maintained for 100 million years. But it's making a particle that can't sustain itself independently of the wasp anymore. So the virus...
But one second. If the virus is endogenized in the wasp, what happens when it reproduces? Is that kept or is it lost? It's not heritable.
You mean when it makes a particle?
Well, at some point the wasp is going to, may breed again.
Yeah. So
is it passed off to its offspring?
Right. So the endogenized virus just gets passed to the offspring, just like every other gene.
It does. Okay.
And it just maintains itself in perpetuity.
But what happens if there's another event like this where the new wasp is, the kids of the original wasp that was endogenized then want to have their own progeny or then become infected, or a parasitoid tries to infect them and they are now immune because they have endogenized DNA previously?
You mean like another virus tried to invade?
Yes, in a successive generation where it's inherited.
Yeah, well, there's actually some evidence that that independent endogenization events have actually occurred. And so one possibility would be that you end up with another endogenization event and it doesn't establish. There's at least one example. There's some suggestion in the literature that maybe in some cases, one endogenization event displaced another endogenization.
That's what I was wondering,
Yeah, and that's kind of what you were getting at. It isn't real clear that actually occurred, but it's definitely been suggested in the literature. And you probably maybe talked with other researchers about endogenized viruses. Endogenization of viruses happens all the time. Most of them just become endogenized and die, so to speak. They just decay, and there's little remnants of them, but they don't really persist for as an intact entity for any length of time. The vast majority of them.
So their purpose is during the life cycle of the creature that they endogenized in, so they don't tend to carry forth into successive generations.
They don't carry forth in successive generations as an intact entity like the ancestral virus was. They just through genetic drift, they just end up accumulating mutations and essentially where there's just little bits and pieces of them over time that are even recognizable. They just decay. That's what happens to most endogenized viruses. What's unique about these polytenoviruses is not only did they endogenize, but selectivists maintained them where they largely function like the ancestral virus, except for the crucial part of being endogenized and not fully packaging all of their genome into particles anymore. In a way, the wasp has domesticated the virus and taken parts of it and used it for its own purposes.
Yeah, I didn't know the relative success of HERVs or other endogenized retroviruses or viruses and other creatures.
Yeah, so
I thought they persisted, but I thought just because the virus doesn't need all of its all of its genes, that it tends to shed them over time to integrate more fully with its host. But you seem to be saying that they're not as successful as I thought.
Right, and most of the time it's just a big dead end, and it's a dead end for the virus. And as far as anyone knows, it doesn't have any value-added fitness benefits for the host either. Retroviruses are really pretty special because of their requirement to integrate as part of their replication process. But countless other bits and pieces of other viruses that aren't known to integrate into their hosts are found in the genomes of all kinds of different host organisms. And how they got there is not very clear a lot of times, but they're there. And they're just usually decayed bits. And even with Nudi viruses in insects, there's a number of examples of endogenized Nudi viruses that have been found through the sequencing of different insect species, in addition to parasitoids. And in most cases, they too are just decayed bits. They haven't been domesticated or co-opted by, in the case of what happened to make polytenoviruses. They're just decayed bits that don't appear to have any utility at all. They're just kind of dead viral pieces in the genome of the host that they happen to endogenize into. And you can recognize them from the fragments. It's not an intact virus by any stretch.
So what's your research about? What are you studying and trying to figure out?
Well, in these kinds of systems, we've spent a good bit of time studying how it is that these endogenized viral elements function. How do they produce particles? How do they package these various virulence traits that are so important to parasitizing hosts? We've spent a lot of energy trying to identify what these various things that get delivered to host insects, these virulence genes that are put into virus particles, what do they do? And why is it that they're so indispensable to the survival of these wasps and enable them, for example, to be a good biological control agent? So that's, at least with the work that we do on pullidoviruses, that's pretty much what we've spent a bit of trying to understand. And then there's
Yeah.
Go ahead.
I was going to say, what useful is there? Anything useful for the medical world?
Useful for the medical world, as in like
that you just for people or for animals. I mean, are there useful techniques or things that you've learned? Vectors to administer medicine, et cetera, drugs.
Right. So one thing that has some utility is some of the machinery that controls the integration of these viral DNAs into wasps. One thing I didn't say is that the viral DNA, the DNAs that get packaged into virus particles that get put into host insects, also integrate into the genome of host insects. And so there's potential for using these things as vectors for kind of gene therapy, but in arthropods, not in people. And so that's a benefit. Some of the genes that make the host insect they prevent the host from being able to develop or be able to grow into an adult insect, or they're very immunosuppressive. These have been used as tools to disrupt immune functions and developmental functions in insects. And so they have potential for use as in a kind of pesticide sort of way, or as an analytical tool for understanding how certain processes work in invertebrate immunology and development. So those are some of the kind of applied ramifications for work on these guys.
Well, very good. What's the best way for people to find out more about your research? Where can they go?
I guess they could go. They could go to a lab website. They could, by typing a person's name in, it's very easy in a Google search to pull up many studies and papers that would describe what I was talking about just now. There are a lot of I think, fairly readable reviews, and not written just by myself, but by other people as well, that discuss the diversity of these organisms and the different variations that I tried to touch upon. So those are very easy ways to kind of gain additional information. And then you're very familiar, I'm sure, with all kinds of databases and the like that you can access that call up literature on a given subject that people are open to any person anywhere. PubMed and other search engines are there for all citizens.
Okay, yeah, very good. If that's the best way for people to find you, that's great. So, okay. Well, Michael, thank you for coming on the podcast. I appreciate it.
All righty, bye-bye.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.